During vigorous exercise, most mammals breathe more rapidly and more deeply. The sound of air moving to and from the lungs can be heard. Listen. In life, the only visible signs of a mammal's respiratory system are the breathing movements of its chest wall and the opening of its air passages at the nostrils. A partial dissection of the body of a sheep can show what the lungs and air passages are like. With this animal, the air passages have been blocked and the body wall already cut through. The lungs occupy this region of the chest cavity. Some cuts have been made so that the chest cavity can be opened and you can see the lungs themselves. The cut begins at the neck, goes under the leg, along the underside, and up along the back leg. If the front leg is pulled back and the body wall, you can see the left rib cage. The ribs have been cut ventrally and also dorsally. If the left rib cage is lifted away, you can see the left lung in the left chest cavity, or pleural cavity. The lungs, partly inflated, occupy most, but not all, of the chest cavity. This is normal. The chest cavity extends to here. You can see here where the ribs of the left chest wall have been cut. The muscular structure here is the diaphragm. It is dome-shaped, bulging upwards into the chest cavity. The stomach and intestines are behind it. The heart lies between the right and left lungs. In this animal, the heart is covered by fatty tissue. It is in this position, here, the lungs themselves are pinkish-white in color. Each lung is partly divided into lobes, with gaps between the lobes. This lung is partly divided in three. This portion, this one, and this larger part. When the lungs are squeezed, they have a spongy feel. Before this dissection began, a closed tube was pushed into the windpipe and air pumped in. This blocked the windpipe and kept the lungs inflated. Watch what happens when air is allowed to leave the lungs. Lungs, in fact, are mostly air spaces. If a piece of lung is cut and placed in water, it floats easily. there is relatively little tissue in it. Lung tissue can be suitably treated, cut into thin sections and stained, then examined under a microscope. Its structure can then be visualized. Seen under low magnification, a thin slice of lung tissue looks like a net. The strands of the net are the lung tissue and blood vessels. The holes in the net are the air spaces. Much of the lung is made up of hollow sacs called alveoli, located at the ends of narrow air passages. As you can see, the air passages have thicker walls. There's one on the left, and another one runs up from the bottom to the right. With high magnification, you can see what the walls of the alveoli are like. Each is very thin and contains fine blood capillaries through which blood flows. The darker reddish blobs are red blood cells. Seeing the main air passages to the lungs requires further dissection. From the nostrils, air travels here and here through the nasal cavities. In the back of the throat, air passes through the same area as the food. Food enters the mouth and moves down past the tongue. The air and food passages cross here. Food then goes down the esophagus. Air passes down through the larynx, then the trachea or windpipe. This is a preserved trachea with parts of both lungs still attached. The trachea itself is a fairly rigid tube held open by cartilage. At its base, the trachea divides into two branches called bronchi, one to each lung. Looking closely at one main bronchus, you can see that it has rings of cartilage in it. Smaller bronchi branch from the main one. Then these branch again and again. There is also an artery and a vein accompanying each bronchus. This is fresh lung, cut open to show the main air passages. The trachea has been cut open at its base. These are the rings of cartilage that support its walls. The rings of cartilage continue down the bronchi. The bronchi continue into the lung tissue. Here and here, dividing and becoming smaller. This is a blood vessel. This is living lung tissue of a dog, seen at the end of an operation at a veterinary clinic. Much of it looks red because it has lost most of its air and collapsed. The red color is due to its blood vessels. Now, watch what happens when the anesthetist pumps air into the trachea.
A little bit more of a puff. There, they've actually been fully expanded. Once more.
This is a chest X-ray of a living dog, seen from above. At the bottom of the picture is the diaphragm. At the sides are the ribs, and in the middle is the heart. Watch the diaphragm first. Inhalation, exhalation. Now the ribs. Inhalation, exhalation. Muscles of both diaphragm and ribs move air in and out of the lungs.
So lots of good stuff in that video. They went through the anatomy really, really well. You could see the cartilaginous rings very well in the trachea and the bronchi. You could see that the lung, although, you know, when he cut through the lung there, it looked like it was solid tissue. When he put it in that beaker of water, it floated because it's mostly hollow spaces. Remember, the lungs aren't like two, big balloons, right? They are millions and millions, hundreds of millions of little tiny air spaces called alveoli. And each one of those alveoli is surrounded by a capillary network, and that capillary network allows for the exchange of gases between the blood and the alveoli. All right, so make sure you're able to trace the passageway of air from our outside world down into our lungs, so know the different tubes. Okay, so if it's in through the nose, it goes through the nasal, the nasal sinuses, nasal cavities, down into the pharynx, past the voice box, the larynx, in past the epiglottis, into the larynx, down the trachea, and then it splits off into the right and left bronchus, and then it gets smaller and smaller into the bronchioles, which no longer have the cartilaginous rings. And then after the bronchioles, it goes down and terminates into the alveoli where gas exchange occurs. All right. Now remember, the lungs are also surrounded by a pleural membrane, and underneath that pleural membrane is a thick band of smooth muscle called the diaphragm. We're going to get into more about how the diaphragm works today. Okay. Any questions at all? All right. So how we breathe has a lot to do with pressure and Boyle's law. Now, I don't know how many of you guys took chemistry and are familiar with that law, but basically what breathing has to do with is the volume of the chest cavity and pressure differences between the inside of the lungs and the outside world. Okay, so I have a few little things. If we were in class, I have this little demo that I would use to show it quite explicitly. We're going to have to try to do our best doing this virtually. But basically what happens is you don't actually suck in air, okay? Air moves all on its own. It moves from the outside world, from the atmosphere, into your lungs because of differences in pressure. All right. So at sea level, the air pressure is 760 millimeters of mercury. Okay, that's one of the ways that we measure air pressure. Okay, the other one is kilopascals. So we're going to use 760 millimeters of mercury. All right, that's our outside air pressure. Now, if the air pressure outside in our atmosphere is greater than the air pressure in our lungs, then air always moves from higher pressure to lower pressure. So in this diagram here, what you'll notice is the diaphragm, okay, the diaphragm is flattening out. So the diaphragm flattens. Okay, so when you contract, the diaphragm flattens, and what that does is it causes the air pressure inside the lungs to become lower. Okay, it increases the volume. So if we were to draw a box around our chest cavity, that represents the volume, which lowers the pressure compared to the air pressure outside, and air rushes in, okay, from higher pressure to lower pressure. Now, when you breathe out, the diaphragm becomes a dome shape, and look what happens to the volume of the chest cavity compared to this picture, right? It's much smaller. So when the volume goes down, the pressure inside your chest cavity, look here, now it's 763. That's greater than the outside pressure, and that's going to force air out. Okay? So air always travels from higher pressure to lower pressure. Okay? Now, how do we affect pressure? By changing the volume. So when you increase volume, okay, when you increase volume, this is a decrease in pressure. And when you decrease volume, this increases the pressure. Okay, so again, the outside air pressure doesn't change. The outside air pressure stays at 760 millimeters of mercury. Okay, that's air pressure. When the diaphragm goes down, so when it contracts, so if you were to take a big breath right now, okay, everybody kind of practice at home. You take a big breath, what you're doing is you are contracting your diaphragm. Okay, and what that means is it's going to flatten, and that's going to increase the size of your chest cavity. So do it right now. Do it at home. Take a big breath and notice what happens to your chest cavity. It gets bigger, right? So we're increasing the volume. And when we increase the volume, that decreases the pressure. Notice the pressure here in our expanded lungs. It's lower than the atmospheric pressure, so air rushes in. Then when we relax our diaphragm, it returns back to this dome shape. That's going to cause the volume to get smaller in our chest cavity, which is going to increase the pressure, and that forces air out. So air kind of moves on its own, right? It moves on its own according to pressure differences. Now, some of you might think, oh, well, what do you mean air pressure? The air doesn't weigh anything. It doesn't exert pressure. Well, look at this video that I'm just showing you right here. This is a tanker from a train. I mean, the metal walls of this tanker are like 3-inch thick steel, right? But what happened is they, with a really powerful vacuum, they sucked all the air out of this train car. And when there's no air inside there, the pressure is really low. It's extremely low. It's like a vacuum. It's almost zero. So the pressure inside that train car is zero. The pressure outside of that train car is 760 millimeters of mercury, just like it is right now with you. But because of the pressure difference, the air just, the air pressure crushes that thing like a pop can. So air pressure is very powerful, and it will move air very quickly. Now, if we think about traveling on an airplane, what they do is they actually have to pressurize the air in the airplane, because if you were way up 30,000 feet in the air, that's kilometers up in the air, there's virtually no air pressure. So you would not be able to expand your rib cage enough to equalize the pressure to allow air to rush into your lungs. Okay? If it was just simply sucking in air, it wouldn't matter about air pressure. But air moves because of pressure differences. So if there was like zero pressure outside, air would never be able to force its way into your lungs. So they have to pressurize the air cabin. Now, if a window blows out or a door flies off or a crack happens in the fuselage of an airplane and all that air rushes out, right? Because the pressure in the cabin is, let's say, 760 millimeters of mercury. Outside the cabin, it's like 0. So all that air rushes out because it wants to go from high pressure to low pressure. And all of a sudden, there's no pressure in the cabin and you can't breathe. So this is why they have these air masks to force air into your lungs under pressure. Okay, so breathing has everything to do with pressure, and how we change the air pressure in our body relative to the outside world is by changing the volume. So if the volume of your lungs of your chest cavity increases, that decreases the pressure and air rushes in. If we squeeze our chest cavity and make our diaphragm go up, that decreases the volume of our chest cavity, which increases the pressure relative to the outside world, and air rushes out. All right, so big star beside these next couple of points in your note package. You have to understand this idea of pressure and volume. And again, that's what's known as Boyle's law, or part of Boyle's law. When you increase the volume, you decrease the pressure. When you decrease the volume, you increase the pressure. Now, how do we change the volume of our chest cavity? It has to do mainly with our diaphragm. Okay, so when the diaphragm contracts, so when that muscle contracts, it flattens. Okay, and it pushes downwards. And that causes the volume of your chest cavity to increase. When the volume increases, the pressure decreases and air rushes in to your lungs from the atmosphere. Okay, so every time you breathe in, you have to flatten that diaphragm. That requires energy, right? You're flexing a muscle. Now, breathing out or exhalation, that's the opposite. So with exhalation, the diaphragm relaxes, goes back to its kind of dome shape. That's going to press up on your pleural cavity, almost like squeezing a water bottle, right? If you were to squeeze a water bottle and the cap really wasn't on all that well, water would shoot out the top. Because what you've done is you've decreased the volume of that water bottle and you've increased the water pressure and water rushes out. That's kind of what happens inside your lungs when your diaphragm relaxes. The diaphragm relaxes, that causes your chest cavity to get smaller in there. That causes the volume of your chest cavity to decrease. That increases the pleural pressure, and air will rush out. So I'm going to warn you, this is a bit counterintuitive. This takes a little bit of sitting down and actually learning this. All right. Hearing me say it is one thing, but if in half an hour I asked you to explain it to me, you might have trouble doing that. So you have to sit down, you have to diagram this, you have to draw it out, and you really have to think about it because it's opposite, right? Pressure increases, or sorry, the volume of your chest cavity increases, that decreases pressure. When you decrease the volume of your chest cavity, that increases pressure. So it's counterintuitive. It's opposite. And I find with these types of these types of concepts, students have a tough time getting it into their brains. So you really kind of have to work at this one. So again, if I were to draw this out, again, I'm not the best artist in the world, but here is my chest cavity. Okay, that's my chest cavity. When the diaphragm relaxes, it does this. Okay, so this is the volume of my chest cavity. And what happens is when the diaphragm does this, what it's done is it's made the volume here it decreases the volume. Okay, all this space underneath the diaphragm now, it doesn't belong here anymore. So what we've done is we've decreased the volume, and what we've done is we've increased the pressure. Okay, so inside here now might be 763 millimeters of mercury. Okay, and outside here is 760 millimeters of mercury. So what's going to happen is air is going to rush out. It always goes from high to low. Now when you breathe in, okay, there's my chest cavity, same size, right? But all of a sudden what I'm going to do now is I'm going to take the diaphragm and flatten it out. Now look what I've done to the volume in here. I've increased it significantly. So the volume increased. And what that does is that causes a decrease in pressure, right? Because there's more room for the air now. So the pressure goes down. All of a sudden the pressure might be now, I don't know, 756 millimeters of mercury. And out here it doesn't change. Our air pressure doesn't change, right? Outside in the atmosphere. So now all of a sudden it's higher outside than it is inside, and air is going to rush in. Is able to change that pressure difference. Okay, now what I mean by that is it's a sealed unit. The pleural membrane that surrounds your lung is sealed. So inside the pleural membrane, not the lungs, but inside the pleural membrane, we don't have movement of gas exchange. Okay, that's a sealed unit. So what we can do is we can change the pressure in that unit. So in that last video, they used that glass bell jar, and they had the balloons inside, and on the bottom they had that rubber membrane. But the glass jar, that represents your pleural membrane. Okay, and your lungs sit inside of that glass jar, right? But inside of there is a vacuum. The pressure does not change. Now, again, if we were in class here, I would show you what would happen if we put a hole in that jar. When I pull down on the diaphragm or push up on the diaphragm, instead of air rushing in through the trachea, air rushes in through the hole in the bell jar. Okay, and then the lungs can't inflate. This can happen in people and in other animals with lungs if we get a hole in our pleural membrane. This is known as pneumothorax, and you might have heard of it called a collapsed lung. So if you're ever hit in the rib cage really hard, and maybe you break a rib and the sharp end of the broken rib punctures your pleural membrane, or if you were in a car accident and a piece of metal flew through there, or if you were shot or stabbed or had some sort of trauma to that pleural membrane, the lung will collapse. Now, what does a collapsed lung mean? It means that when you try and change the pressure in that pleural membrane, instead of air rushing in through your trachea and down your bronchi and all that kind of stuff, bronchioles, what happens is air would actually rush in through the wound, And if air gets into the pleural membrane, it causes pressure, and that basically causes the lung to just collapse in on itself. So a collapsed lung is very serious. And here you see an X-ray of pneumothorax. You can see that the right lung on the left side of your screen is inflated, right? That's the black structure. And the left lung on the right side of your screen, you can see that it is not inflated. It's completely collapsed. And that's because there's some sort of hole in that pleural membrane that's causing air to rush in and collapse that lung. Now, when you are exercising and you need an increased amount of oxygen, so your muscles are demanding more oxygen because they're working harder, you're also making more carbon dioxide. You need to get rid of that carbon dioxide quicker. You're going to breathe, your breathing rate is going to increase. And also the depth of your breathing. So you're going to breathe a lot deeper. And when this occurs, your diaphragm can't do it all by itself. So during times when you have to breathe deeper and more often, muscles in between your ribs help to draw your rib cage upwards and downwards to increase and decrease that pleural pressure more often and more severely. These muscles that are in between your ribs are called your intercostals, and you have two sets of them. You have an internal intercostal and external intercostal muscles. And again, these are found in between your ribs, okay, your actual bones of your ribs. So the internal intercostal muscles pull your rib cage downward, and your external intercostals pull your rib cage upwards when you need to breathe deeply or quickly breathe in. Now, these muscles are different than your diaphragm. Okay, remember your diaphragm is made of smooth muscle. And smooth muscle, if you'll recall, is involuntary. It forms sheets and tubes. It's found in your organs. It's found in your digestive tract. It's found in sphincters. It's found around your arteries, that sort of thing. Okay, it's got one nucleus. It's flat sheets. Now, the muscles found in between your ribs, these are a different kind of muscle, and these are called skeletal or skeletal muscles. Okay, and they're a little bit different in structure and form. Now, if we look at skeletal or skeletal muscles under a microscope, you'll notice that they look like they have stripes. Okay, those stripes are called striations. So one of the first clues when you're looking at muscles under a microscope, what type of muscle you have, is to look for these stripes. So these are known as striations, and we'll talk about why skeletal muscles have stripes like that later when we look at the actual structure of the muscle. The other thing that I want you to notice is that's one muscle cell there, and you'll notice that there's 3 nuclei. So I know in Science 10 and in junior high when you studied the cell, you were taught that a cell has a nucleus, right? That's it, one nucleus. But that's not always the case. So here is a cell in our body, and it actually has multiple nuclei. Now, we talked about certain cells in the body. This is kind of going back just as a review. We talked about certain cells in our body that don't have any nuclei at all. Can anyone remember an example of a cell in our body that lacks a nucleus? Yeah, red blood cells. You got it. Excellent. So red blood cells are a type of cell in our body that don't have any nucleus at all. I'm surprised nobody said the other one, which are the platelets, the thrombocytes. They don't have a nucleus either. But yeah, RBCs. So some cells don't have a nucleus at all. Other cells, like skeletal muscles, have multiple nuclei. All right? Now, where do we find skeletal muscles? We find them, this is the meat of a body, okay? If you sat down and ate a steak, a beef steak, that's the skeletal muscle of a cow. If you sat down and ate some baby back ribs, those are the skeletal muscles of the pig, right? And actually, they're the intercostals, the muscles found between the ribs. So these are skeletal muscles. Now, skeletal muscles are striated. They have multiple nuclei per cell, and they are voluntary. So you have control over them, but they are different from skeletal muscles. So the internal and external intercostal muscles are skeletal muscles. So they help us to breathe. But skeletal muscles also have all sorts of other functions. So they allow our joints to move, right? If you were to bend your arm right now and scratch your nose, your elbow is articulating. Okay, it's moving. It's a joint. Now, how come that elbow is moving? It's because you have a bicep muscle attached to one side of it, and when you want to release that joint, okay, and straighten it back out, you have another muscle on the other side called your tricep that's going to pull your elbow straight. So muscles, when they work in pairs, they allow our joints to move. Okay, they support our body. They enable us to stand upright and to move. They provide protection. So your abdominal muscles provide a lot of protection for your internal organs. Muscles are attached to tendons, and then tendons are attached to bones, and that helps to stabilize our joints. Okay, and as well, muscles, skeletal muscles, they can generate a lot of heat. So what's one of the first things you notice when you're getting really cold? What happens? So you kind of put your hands underneath your armpits, you try and decrease your surface area of your skin. But if it's getting really cold and you're getting really cold, how do you know it's really time to get inside? Well, you start to shiver, right? And what shivering is it's your skeletal muscles contracting and relaxing very quickly. And the reason for that is because you want to generate a bunch of heat. And as I mentioned before, skeletal muscles are the most metabolically active tissues in your body, meaning they use up a tremendous amount of energy. And when they're using up that energy and burning that energy, they are releasing a tremendous amount of heat. Okay. Now, unlike smooth muscles, skeletal muscles can only work when they are contracting. Okay, so skeletal muscles only do work when they are contracting. And what I mean by contracting is shortening. Okay, so if I were to ask you right now to flex your bicep, make a muscle, okay, the gun show, what you would notice is that your hand moves up towards your shoulder, and that bicep muscle gets shorter. Okay, sorry, your hand moves up towards your shoulder, and the bicep muscle shortens. Okay, it can do work. So when you're doing a bicep curl, you've increased the resistance by putting a weight in your hand, and what you're doing is you're overcoming that gravitational force on that weight, and your muscle has to work really hard. But when you're doing a bicep curl, your triceps aren't working. Okay, now when you lower that weight, or if you were to extend your fist away from your face, away from your shoulder, down toward your hip, you'll notice that the muscle at the back of your arm, your tricep muscle, flexes, and your bicep relaxes and lengthens. So skeletal muscles only work when they pull, not push. So because of that, every joint in your body is controlled by a pair of muscles that work in opposition to each other. So again, your elbow, think about your elbow. When you I don't know, cause the angle of your elbow to become less, right? Your bicep is working. When you increase that angle, your triceps are working. Think about your when you're sitting down, and if you were to pull something towards you, you're using the large muscles in your back, right? And then if you were to push something away from you, you're using the muscles in your chest, your pectoral muscles. So your latissimus dorsi, those are the big muscles in your back, and your pectoral muscles, the muscles in your chest, they work antagonistically. All right? Think about the big muscles in your leg. When you kick a soccer ball, you're using your quadriceps. Okay? When you bring your leg back to get ready to kick that soccer ball, you're using your hamstrings. Okay? Those are the muscles kind of in your back. All right? So every muscle, a skeletal muscle that's attached to your skeleton, has a partner that works in opposition. So these are known as antagonistic pairs. So an antagonist means it's the opposite. It's working in opposition of. So every time a joint moves, even the little joints in your fingers, the joints in your wrist, all of them are attached to separate muscle groups that work in opposition of each other. Okay, and this is known as antagonistic. So back to breathing, we have internal intercostal muscles, which pull our rib cage downwards. And then we have external intercostal muscles, which pull our rib cage upwards. So they work in opposition of one another. That's when we're breathing in, our internal intercostals will flex and pull our rib cage inwards. And when we're breathing out, our external intercostals will flex and pull our rib cage outwards. Now, one of the places that students have the most trouble in this chapter is this particular area right here, the organization of a muscle, a skeletal muscle. There's lots of strange words here, and there's lots of anatomy to learn here. And a lot of the words kind of sound the same. So I'm going to kind of go through this fairly quickly, but what you guys are going to have to do is you're going to have to go back after and you're going to have to kind of learn this stuff by either diagramming it or reading your textbook. I'll also give you a little bit of homework that you need to do. So this is important, and it's also a little bit tough to learn. Okay, so you're going to have to put a little bit of effort in here. So if we were to take a muscle like your bicep, for example, your bicep is actually made up of muscle cells, okay? And we call these muscle cells, we call them muscle fibers. So a muscle cell and a muscle fiber, same thing. All right, so that's number one, that's a place of confusion. So when I'm referring to a muscle fiber, what I'm actually referring to is one muscle cell. Now, muscle fibers or muscle cells are bundled together to form a unit called a muscle fiber bundle. All right, so look at the diagram I have here on the screen. And again, this is hard for you guys because in Science 10, you learned that a cell looks a certain way, right? You were given a diagram of a cell. An animal cell looks kind of round. It's got one nucleus and a cytoplasm and Golgi bodies and all that kind of stuff. You've never seen a cell that looks like this. This thing right here that's highlighted, that is a muscle cell. That's one muscle cell. Okay? Now, inside the cytoplasm of that muscle cell are a bunch of proteins, and we're going to talk about those in a second. All right, so the basic unit of a muscle is a muscle cell, aka also known as a muscle fiber. Okay, so a muscle fiber and a muscle cell are the same thing. Now, if we take a bunch of muscle fibers and put them together, so here's another muscle fiber right here. Another muscle fiber right here, and another one 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 right here. Those are all individual muscle cells. And if we put them all together, that's called a muscle fiber bundle. Okay, so this thing right here is a muscle fiber bundle. If we take a bunch of muscle fiber bundles and put them together, then we get a muscle. Okay, so your bicep is a muscle. Your bicep is made up of thousands of muscle fiber bundles. Each muscle fiber bundle is made up of individual muscle fibers, which are muscle cells. All right? Now, muscles are surrounded by a thick connective tissue called a tendon, and then the tendon gets anchored to a bone. And that allows the joint that bone belongs to move. All right? So it goes bone, tendon. The tendon wraps a muscle, like the bicep. The muscle is made up of muscle fiber bundles, and the muscle fiber bundles are made up of muscle fibers. So you should be labeling the diagram on page 7 of your note package. Okay, so you can see it's a little bit confusing already, and we're not done yet. Now, if we were to take a look at one muscle cell, at an individual muscle cell, okay? So this thing right here is one muscle cell. Remember, fiber equals muscle cell, right? So that's one cell. Now, in the cytoplasm of the cell, okay, you have one of your nuclei. Remember, they have multiple nuclei. So here's one nuclei. You have what's known as the sarcoplasmic reticulum. That's basically the endoplasmic reticulum. And then you have a cytoplasm. They call it the sarcoplasm, but it's just a cytoplasm. And inside that cytoplasm are proteins, long, thin proteins that are called myofilaments. There's two types of myofilaments. There's actin and myosin. And these myofilaments are bundled all together into something known as a myofibril. And then the myofibrils are bundled all together to make up a muscle fiber. And then the fibers are bundled together to make a muscle fiber bundle. And then the bundles are bundled together to make a muscle. I know some of you must be just face palming right now, right? Like, oh my God, what the heck is going on here? There's a lot to learn here. All right, so if we go from smallest to largest, the smallest structure of a muscle, these are little tiny protein wires called myofilaments. That's the smallest part of a muscle. If we take these myofilaments and bundle them all together, that makes up a myofibril. If we take a bunch of these myofibrils and bundle them all together, we get a muscle cell or a muscle fiber. If we take a bunch of these muscle fibers and bundle them together, we get a muscle fiber bundle. And then if we take a bunch of muscle fiber bundles and bundle them together, then we get a muscle, like your bicep. So again, at the bottom of page 7, I kind of have a flow chart there On the left-hand side, if you want to just put on the left-hand side of that flow chart on the bottom of page 7, on the left-hand side where it says muscle, that's the largest unit. So put it as an example there, a bicep. And then on the far right-hand side, the myofilament, that's the smallest part of a muscle. Okay, those are the little protein wires that are found in the cytoplasm of a muscle cell. Now, also bundled up in the muscle are nerves that come from your spinal cord that also come from your brain. Okay, and those nerves allow you control over those muscles. And also, there's a blood supply there, right? We need to have lots of blood going to these muscles to supply it with sugar and oxygen and take away carbon dioxide. Okay, the myofibril is made up of these little tiny protein chains called myofilaments. And there's two types of myofilaments, and we'll talk about them in a second, actin and myosin. Okay, so the myofilaments form the myofibrils. The myofibrils are found inside the muscle fiber. We put a bunch of muscle fibers together to make a muscle fiber bundle. Then we put a bunch of muscle fiber bundles together to make a muscle. All right, so here's that flow chart. So as far as biggest goes to smallest, this would be the largest structure. And this would be the smallest. Okay, so these two are found inside the cytoplasm. Okay. Muscle fiber, another word for that is a muscle cell. And then these muscle cells are all bundled together to make muscle fiber bundles. And then muscle fiber bundles are bundled together to make muscles. Okay. So just one more look at it. So again, we're looking here at this. I think that's called the brachialis muscle. I'm not sure, man. Anatomy was a long time ago for me, but I believe that's the brachial radialis muscle. And if we look at that muscle, you can see that's made up of all these muscles, right? If we take one of them and look at it, it's surrounded by a tendon at the base. That tendon would be attached to the elbow bone, okay? And now if we looked at the muscle itself, what we would find is the muscle is made up of these muscle fiber bundles, okay? Bundles of muscle fibers. Now, what is a muscle fiber bundle made up of? A bundle of muscle fibers. And again, remember, muscle fiber is the same as a muscle cell. Now, what's found inside the cell? Myofibrils. And what are myofibrils made up of? Myofilaments. Now, how does a muscle contract? What happens? Well, it has to do with the two smallest units that we find inside the muscle cells, the myofilaments. And I mentioned there's two myofilaments. There's what's known as actin and myosin. And actin and myosin are made up of protein. So when you see these people walking around, these bodybuilders that are trying to build muscle, and they're walking around with their blender bottles filled with protein powder, the reason why they're increasing their protein intake is because what happens when you build muscle is you rip apart these little tiny myofilaments. So you break them down, you actually damage them, and your body has to rebuild them. And when your body rebuilds them, it makes more of them and it makes them thicker and stronger, and that requires protein. So actin and myosin are fibers. Think of them as ropes, but they're ropes made of protein. Okay, and they fill our muscle cells. And when they interact with each other, they cause your muscle to shorten and exert force on the joint. Now, these two myofilaments, actin and myosin, they sit next to each other. So think of an ice cream sandwich. You guys all know what an ice cream sandwich is, right? It's got, it's got like a cookie on top, and in the middle, it's got some vanilla ice cream, and then it's got, another cookie on the bottom. So actin and myosin are kind of like that. Think of the actin as the cookie in the ice cream sandwich, and think of the myosin as the ice cream. Okay, so the myosin kind of sits in the middle, and the actin sits on either side. And when these two interact with each other, they kind of slide. And what that does is it causes the muscle overall. If they're all the actin and myosin are doing this simultaneously in a muscle fiber, then the muscle fiber will actually shorten, okay, and exert a force. So the actin is thin. It's what's known as the thin filament, and it's when we're talking about muscles contracting, we think of it almost like a rope. Myosin is known as the thick filament. And myosin is not really like a rope. Myosin is rough, and it's got all of these little kind of like, I don't know, paddles on it. Okay, so each myosin molecule has millions of these little heads that look like a paddle on top. And these heads can go back and forth. Okay, these heads are not fixed. They can rotate. They can actually move this way, and they can move this way. And what they do is they grab the actin like hands would grab a rope, and they can cause the actin to slide. Okay, so you have to picture, like I said, like an ice cream sandwich, you would have to picture that there would be an actin molecule here like this, and an actin molecule kind of down here like this. And what would happen is these heads here, they would attach to the actin, okay? And because these heads can move back and forth, they would cause the actin to move back and forth. And this would shorten and lengthen your muscle. Now remember, this is all happening in the cytoplasm of the muscle cells. Okay, so here's a much better diagram than the one that I just drew. But what you can see here is you can see that the actin sits on either side of the myosin. So again, up here is actin. Right here is your myosin. And then we would have another actin here. And if we would continue on up and down, we would have another myosin, another actin, another myosin, another actin. There's thousands and thousands of these in each muscle cell. So when it's time to contract your muscle, so again, if you are, you're sitting in your chair, or maybe you're lying in bed or whatever, your hand might be resting. If you were to all of a sudden bring your hand up to scratch your nose, your bicep just shortened. So when your biceps shorten, what that entails is that in each cell inside that bicep, the myosin has to grab the actin and pull that actin so that it shortens the muscle. So the actin is attached to these bands called Z bands, and this is what gives muscles the striated look. So the reason why muscles look like they have stripes to them is because of these Z bands. So the actin is anchored to the Z band, and when the myosin pulls on the actin, it pulls on the Z band and it causes the muscle to get shorter. Okay, so kind of think of it like this. This is my myosin, okay? And the myosin has these heads. So think of the arms as like myosin heads. And these heads can attach to the actin. And when we contract a muscle, what happens is the myosin pulls the actin, and that causes these Z bands to come closer to each other. And that's going to shorten the muscle. So the myosin pulls on the actin, and the actin is anchored to the Z bands. And as the Z bands get closer, that causes the muscle to become shorter. So it's the myosin that's doing all the work. Yes, Jada, that's known as isometric work. So what's happened is when you're holding your phone up, or if you were to do like a wall sit against a wall, right? You're not actually causing the muscles to shorten, but you're causing them to stay at a halfway shortened point or at a full shortened point when you have your phone up to your hand, and that requires energy. So they would stay. You're absolutely right. They would stay curled up. They would stay contracted until you actually put your phone down, or if you were doing a wall sit, until you actually stood up. Okay, so that's an isometric type of a movement. Your muscles are still working really hard to maintain that, but they aren't in the active phase of shortening. They're just held at a certain point. So this idea of myosin grabbing actin and pulling the actin, which pulls on the Z bands, which causes the muscle to contract. This is known as the sliding filament model of muscle contraction. Now, what's important here in the sliding filament model is, of course, you have to understand the anatomy. So I'm going to be introducing you to a number of different proteins that are involved here. But equally as important is you have to understand the steps that are involved in order to shorten a muscle. Okay, so all of a sudden, yeah, that's great. Let's use a phone as an example, right? Your phone is on the desk and you're standing by it. All of a sudden, you get a text message. So you want to look at it. You grab your phone and you flex your bicep to bring your phone up to your face. All right, so the contraction of a muscle begins by flexing the myosin head backwards. Okay, so the first thing that happens is you get a nerve impulse to your muscle, and that nerve impulse tells your muscle that it's going to contract. And what needs to happen are these myosin heads need to be flexed. Okay. Now, the example that I always use is a mouse trap. I think everybody's used a mouse trap before, right? So here you have the base of your mouse trap. Then you have a spring right here. And then you have that bar that's going to kill the mouse, right? So think of the myosin like the bar, the myosin head like the bar of the mousetrap. In order to get the mousetrap to work, we need to take and pull this bar backwards, and that requires energy. All right, in order to pull that bar of the mousetrap backwards, so this is a mousetrap, okay? This is a spring in the mousetrap in the middle, this is the wood part, and this is the metal part, right? So in order to pull that metal bar backwards, you are exerting energy in there. And then if you were to let that metal bar go, it would snap, right? Think of that metal bar on a mousetrap like a myosin head. When the myosin head is in this position, it's not ready to do anything. So what we need to do is we need to bring it backwards, like the bar of the mousetrap, to get it ready to do work. And in order to pull that myosin head back, that requires energy. Okay? So the myosin head has to be brought backwards. Then what it's going to do is it's going to attach itself to the actin. And then once it's cocked and attached to the actin, it's allowed to spring forward like the mousetrap springing forward. And that shortens the actin by a little bit. Then the myosin head would be cocked back again, would grab the actin in another position, and then fire forward and shorten the actin. So these little paddles, these little myosin heads, they're working thousands and thousands of times in a split second. They go back, they grab the actin, they go forward. They go back, they grab the actin, they go forward. And little by little, this is going to shorten the muscle. Okay? Now, anytime we use energy in our bodies to do an activity, this requires ATP. Okay, now ATP stands for adenosine triphosphate, and ATP is like a battery pack for our cells. So anytime our cells need to do anything with using active transport, like we talked about in the kidneys, or anytime the cell needs to move, or parts of the cell, like the myosin head, have to move, this requires energy, and that energy is in the form of ATP. Okay, so in this diagram here, think of this as the mousetrap that's just kind of sitting there, right? We want to bring that mousetrap arm backwards. We want to bring that myosin head backwards. We want to bring it to the ready position. That requires ATP. Just like you have to put energy in to flex that spring in the mousetrap to get it set, your cell needs to exert energy on the myosin head to get it into the ready position. So this outline right here, that's where we want the myosin head to be. In order to get it there, that requires ATP. Okay, so this is flexed. This is relaxed. In order to get it flexed, we have to input ATP. Now, we haven't talked about cellular respiration yet, but ATP is made in cellular respiration. And cellular respiration is done in the mitochondria of our cells. So what we find is in muscles, we have lots and lots and lots of mitochondria. Remember, they're the powerhouse of the cell. They are responsible for making our ATP. Now, ATP can be made in two different ways. It can be made with oxygen or without oxygen. Now, with oxygen, we make way more ATP in our cells than we do without oxygen. Okay, so when we're making ATP with oxygen, this is known as aerobic respiration. Okay, anaerobic respiration, this means without oxygen. So it's about 6 times more, we get about 6 times more ATP out of a molecule of sugar if we burn it in the presence of oxygen than we do if we burn it without oxygen. So it's way more efficient for us to burn sugar in the presence of oxygen than without. Okay? Now, another thing that happens when we burn sugar without oxygen, first of all, we don't get a lot of energy out of that sugar. Second of all, we make byproducts that are toxic to us. And one of those byproducts is lactic acid. So those of you who are athletes out there, you guys know exactly what I'm talking about, right? I played a lot of soccer when I was younger, and I played hockey up until a couple of years ago. So I'm familiar with those two sports. And you know, if you are going out on a shift in hockey and you are going as hard as you can only last really about 30 seconds before your muscles are ready to cramp and give out on you, right? And that's because what's happening is you're not delivering enough oxygen to those muscles, and they are not burning the sugar that you are supplying them with very efficiently. So you're not making a lot of ATP, which means that eventually the muscle is going to run out of energy and it's not going to be able to contract and relax anymore. Also, you're building up a bunch of lactic acid, and that's going to interfere with the way the muscle works. And eventually, what it's going to do is it's going to cause cramping, and your muscles just won't work. Okay. Again, those of you who might not have played, those sports, but you might be active in the gym, if you were to rack up your max weight on the bench press, you might only be able to do it 2 or 3 times, and eventually, your muscles are going to crap out on you, and you're not going to be able to lift that weight anymore because you don't have enough energy, right? So, aerobic activities are activities where you are supplying enough oxygen to your muscles where they can burn the sugar efficiently and you can go forever. I had a friend who ran ultra marathons. So just to put that in perspective, a regular marathon, I want to say around 42, 45 kilometers, something like that, right? He would run like 140-kilometer marathons. It would take him all day. He would literally run for 24 hours. And I remember being at a water station. I went to support him one day. He was doing one of these, and he was barely breathing hard. Okay. And the reason for this is because he was in really good shape, but he could supply his muscles with enough sugar and enough oxygen where they were getting enough ATP and they could go on forever. Right? Now, conversely, I don't care how fit you are. My friend who ran ultra marathons, I played hockey with him. His shifts were 30 to 45 seconds, just like everyone else, because that is what's called anaerobic activity. So you're working so hard that you can't supply your muscles with enough oxygen to burn that sugar efficiently, and eventually they will crap out. And usually when you're doing anaerobic activities, you can only do them for, a minute or so before you can't do them anymore. All right. So this is why I grouped the respiratory system and the muscle system together in one node package. Number one, muscles are really important in breathing, right? Our lungs have no muscle, they don't breathe. It's the surrounding muscles that are important in breathing. And number two, one of the main reasons we need oxygen is to supply muscles so that we can have aerobic cellular respiration so that our muscles can work efficiently and with enough ATP to allow them to work. So our circulatory system is really important as well because our circulatory system allows us to deliver that oxygen and get rid of the CO2 and the heat that is created when our muscles are contracting. So in addition to ATP, cellular respiration makes lots of carbon dioxide and heat, and that has to be taken away from the muscles And that's done through your circulatory system and your respiratory system. So all these systems are intertwined. Talking about all these different systems, and we're talking about them in these these chapters, these separate chapters. But really, if you think about it, they are all interdependent on each other. The respiratory system is dependent on the circulatory system to deliver oxygen and bring CO2 to the lungs, right? The muscular system is dependent on the circulatory system. Also dependent on the digestive system to make sure we get enough sugar for energy. So it's all interdependent. Now, again, ATP is really, really important because ATP allows for those myosin heads to be brought back in order to grab the actin and pull the actin. Without ATP, the myosin heads cannot work. And if the myosin heads can't work, then the muscle cannot contract. Now, we have a bit of a backup in case we run out of ATP, and the backup is called creatine phosphate. Now, ATP is like a rechargeable battery. So ATP stands for adenosine triphosphate. Okay, and the way ATP works is that what happens is ATP looks like this. It's got an adenosine head, and then it's got 3 phosphates that are attached to it like this. Okay, hence the name adenosine triphosphate. So the way adenosine triphosphate works is what happens is we bust off this 3rd phosphate molecule, and ATP turns into something called ADP, okay, which stands for adenosine diphosphate. All right, only 2 phosphates at the end. So when this 3rd phosphate gets broken off, this is where we get energy from. So an ADP molecule doesn't have any energy to do work. So what we need to do, and what the mitochondria does, is it reattaches, it adds a phosphate, and it causes the ADP to become ATP again. So it recharges it. Now, this happens in the mitochondria all the time. But what if you are exercising really hard and you don't have time for your mitochondria to make new ATP? Well, what happens is we have a small supply of this stuff called creatine phosphate. And creatine phosphate has a spare phosphate on the end of it. And what creatine phosphate can do is it can lend that phosphate molecule to the ADP and make it become ATP again. Now, the problem is we don't have very much of this creatine phosphate, only a few seconds worth. It's kind of our backup emergency reserve. So what people have done who want to work harder in sports is they will take artificial creatine. Okay, so it comes in the form of a powder, and you can mix it in a drink, and you can drink it down, and you will absorb this into your bloodstream before you work out, let's say, or before you were to go to a basketball game or a hockey game or whatever. You could take this stuff, and it would give you an increased supply of creatine phosphate. And what that would do is it would give you a little bit of extra time to work before your muscles kind of crapped out on you. So this is not going to make you stronger. Okay, it's not testosterone. It's not going to make your muscles bigger. It's not human growth hormone. It's not going to make your muscles bigger. It's not going to make you burn energy. If you were to just drink a bunch of creatine powder and sit in your desk or sit on your couch and do nothing, it would do nothing for you. What creatine powder, what creatine supplement does is it gives you a little extra boost when you're performing an activity. So instead of doing 10 reps of bicep curls, it might let you do 15. Okay, so it allows you to work harder. Now, there are many downsides to taking creatine supplements. It's very hard on your liver because you need to break down this extra stuff that you've put in your body. It's hard on your kidneys because your kidneys have to get rid of this creatine. Okay, you have extra creatine. It causes your tissues to become hypertonic, which means that you'll retain water, so you look really puffy, and again, that's kind of hard on your kidneys. Okay, so there are many dangers to taking creatine phosphate supplements. And in my opinion, if my son were to come to me and say, "Hey, Dad, you think I should take this creatine?" I would say, absolutely not. All right, now you might have trainers, if you're a high level athlete, you might have a trainer that it recommends this stuff for you. But for your average person, your average weekend warrior, the average person who's going to the gym, you don't need this stuff. Okay, there are more dangers than there are benefits. Now, again, if you have a professional who's monitoring you, your dosages and all that kind of stuff, then maybe. But personally, I wouldn't let my own kids take these supplements. Okay. Now, the sliding filament theory involves the myosin heads grabbing the actin and pulling the actin, which is attached to those Z lines, and that's going to shorten the muscle. But there's a little bit more involved in that, right? We have to have some control over this. We don't want your muscles just working at random. So how is the contraction of your muscles controlled? Well, it's controlled by an auxiliary set of proteins that surround the actin filament. And when your muscle is resting, when you don't want to contract your muscle, the actin is blocked from joining the myosin. Okay? So there are guards that are set up around the actin that prevent the myosin from just randomly attaching to the actin. Now, sometimes this happens and you get a muscle spasm, right? I mean, I think we've all had that, where all of a sudden your muscle will just start to contract and relax just in one random place. Okay, that's what would happen all over your body if we didn't have this control mechanism. Now, the guard that blocks the actin from binding to the myosin is called tropomyosin. Okay, so in the diagram up on the screen right now, the actin is in green, the myosin is in, what is that, violet, I guess? And then the tropomyosin is in blue. And you'll notice that the tropomyosin is in the way of the actin and myosin binding. So it's kind of like a guard. Now, as soon as you want to contract a muscle, you have to be able to allow the actin and the myosin to join together. So we need to get the tropomyosin out of the way. So at rest, the tropomyosin blocks the myosin and the actin. Now, when it's time for a muscle to contract, a nerve impulse goes down into your muscle and it causes calcium ions to be flooded into your cytoplasm of your muscle cell. And the calcium joins up to a special binding site called troponin. So this thing right here is troponin. This thing right here is called tropomyosin. Okay, now tropomyosin is like the guard, and troponin is a binding site for calcium. Now, when calcium binds to the troponin, It causes the tropomyosin to change shape, and the tropomyosin gets out of the way, and myosin can now bind to actin. Every time a muscle contracts, whether it's your eyelid blinking or your finger twitching or your bicep contracting or the muscles that are involved in allowing you to walk across the floor, every time a muscle contracts, no matter how little or how much, this has to occur. A nerve impulse has to go from your brain through your spinal cord to the muscle fiber, to the actual muscle cell. Okay, that nerve impulse causes calcium to be released into your cytoplasm. The calcium flows very quickly and it binds to the troponin. The troponin, when it's bound with calcium, causes the tropomyosin to change shape and get out of the way of the actin. And now, all of a sudden, the myosin can grab hold of the actin. And if the myosin has been contracted using ATP, it'll then grab hold of that actin and pull it. Now remember, the actin is attached to the Z lines. As the myosin pulls on the actin, that causes the Z lines to get closer together, and that shortens the muscle. Now, as soon as you want to relax the muscle, we need to get rid of all that calcium. So there are pumps that grab that calcium and take it out of your cytoplasm really quickly. As soon as the calcium is taken out of there, the tropomyosin goes back to its guard position, and now the myosin and actin can't bind and the muscle is relaxed.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
So this video did a very good job of showing what happens during that cross bridge formation. Okay, what we've referred to as the sliding filament theory of muscle contraction. So without calcium, none of that occurs. All right. So we get a signal from our brains through our nerves. Okay, that signal goes right down to the individual muscle cells. And when the individual muscle cell gets the activation from our nerves, it dumps calcium that's stored in what's known as the sarcoplasmic reticulum, what you guys might know as the endoplasmic reticulum. Okay, there's tons of calcium in there. And as soon as your muscle cell gets a message from a nerve cell, it dumps that calcium into the cytoplasm. Now that calcium diffuses immediately and binds up with the troponin molecules. The troponin molecule, when it's bound to calcium, changes shape, and that causes the tropomyosin to get out of the way of the actin binding sites. As soon as those actin binding sites are exposed, the myosin, which has been kind of cocked like the mousetrap, right? Ready to go. That myosin head grabs a hold of the actin and then it fires forward. And little by little, if that happens over and over and over again, okay, the actin, which is attached to those Z lines on either side of the muscle cell, causes those Z lines to come closer together, and that shortens the muscle. Now, if you want to relax the muscle, we have to get rid of the calcium. So there's almost like a calcium vacuum, these calcium pumps. They actively transport all that calcium out of the cytoplasm back into the endoplasmic reticulum, sarcoplasmic reticulum. Okay, and then we get rid of all that calcium. The troponin goes back to its regular shape. That slides the tropomyosin over the actin, and then the myosin can't bind and your muscle is relaxed. So this is a very complex procedure, but it's happening all the time, constantly, when your muscles are contracting and relaxing. Okay, so this kind of shows it again. So that big red thing that's going back and forth, you guys know what that is? Can you think of what that is? Okay, that's the myosin. What are the golden kind of look like a pearl necklace there, intertwined, those golden balls in that big long chain? That's the actin. Notice how the myosin head is binding to the actin. Now, what would the blue rope be there? That's the tropomyosin. And then the purple set of shapes there on the blue, that would be the troponin. Okay, notice how the tropomyosin slides out of the way, and that would be the result of calcium binding to the troponin. That causes the tropomyosin to slide out of the way. All right, so there is a ton of stuff here. There's a lot of complicated material. I really went overboard here today. This was a long lecture with a ton of material. All right, so I need you guys in your textbooks to read pages 332 to 342. Then in your note package on page 11, I've put a number of questions that I would like you to work on. Okay, so in your note package, page 11, there are a number of questions that I've put in there that I would like you to work on.